All right, why don't we go ahead and get started with our last session for today, which will be chaired by Jennifer Lippincott-Schwartz from Janelia.
Great. So this session is entitled Beyond Connectomics, Metabolism-Driven Brain Physiology. We're really excited to now sort of shift into the brain and the role of metabolism in the brain. We know a lot about how neurons are connected to each other to create synaptic pathways. What we're going to be focused on in this session is how metabolism can facilitate or hinder that whole process. So the three people who are going to be talking in this area are Meng Wang from Baylor College of Medicine, Yellen from Harvard University, and Daniel Hochbaum from Harvard Medical School in the Sabatini lab there. So let's get started. Meng, why don't you share your slides and present your thoughts.
Can you see my screen? Okay, great. Yeah, thank you, Jennifer, and also Wamsi and Ron for organizing this exciting workshop and to learn all different aspects related to these topics. So in my 10 minutes, I will like to discuss metabolic imaging and profiling in vivo. So not necessarily restricted to brain, but I think just overall. And because I think technologies in these areas will be required for study 4D cellular physiology. And I also think current expertise and Janelia can help drive advances in this area. So ultimately, I think those new development will benefit the whole community. So, think about the connections. So actually, when I first read Louis Sullivan's code, form, follow functions, and I saw living organisms actually the best architecture designs. So they have structures and functions always correlated at all levels, from subcellular organelles to cells and to multicellular tissues. And so unlike in Petri dish, if we look at in vivo and in live organisms, actually those different structural units are not isolated. So they are communicating and they can form direct interactions like ER mitochondria membrane contact or synaptic junctions. And there are also indirect interactions between them using paracrine or endocrine signals. So through those communications, those different structure units can coordinate their functions and to support physiologies in organisms. But those communications are not fixed in one state. So they undergo changes with time and in different environment. So new connections can be generated during macrophage infiltration into adipose tissues. And existing connections can be also lost, like during the neuron degeneration process. Then the question is, what can drive those changes in communications? So one crucial input is from metabolism. So when metabolic status in one of those units change, then the communication can be formed or lost. So therefore, the metabolic flux in those different units can really shape the communication network. And in addition, I think we heard from several talks, the metabolite can also serve as signals to mediate those communications directly. So the example I like to share with you is from C. elegans, our model system. So in intestine is their major fat storage cells, and it's not innervated, but the induction of lysosomal lipolysis in the intestine can drive the secretion of polyunsaturated fatty acid and to act on neurons. So how this works, so actually those polyunsaturated fatty acid, the lipid metabolite can act as a ligand for nuclear hormone receptors in neurons and then upregulate neuronal peptide expression together. So it may lead to the overall changes in neuromodulation in the nervous system. So from this example, you can tell that the metabolite or metabolic signals actually can help establish a long-range communications between two tissues that are not directly connected. So therefore, I think beyond mapping structural connections between those units, to build a connectome, we also need to map their physiological connection. I think this is the 4D cellular physiology 4. And then the key step is to image or visualize metabolic changes across different scales in time and space. So for this, we'll need technologies that can help us track metabolic dynamics at all levels in vivo. So we currently have some technologies available. So for example, in my group, we use hyperspectrum stimulated Raman scattering microscopy to image metabolic changes in vivo. So this technology takes advantage that different chemical bonds has different vibrational energy. Then it can generate specific signals when the two laser energy difference match with the vibrational energy of a chemical bond. So this way we can change the lasers across a series of different wavelengths, then a special SR spectrum can be derived from the combination of a chemical bond in samples. So for example, you see here the triglyceride versus cholesterol ester, so they can give distinct SR spectrums. So making it possible for us to image them separately at a single lipid droplet resolution without any labeling. So this chemical method then is non-invasive and then can be used in live animals. So using this platform, then we can image live worms and then to acquire metabolic features. So the SR spectrum, as shown here, is deconvoluted into 4 components. So then for different cells, we find the signal intensity is different for each different component. We can then integrate all those information from 4 different components together and to generate a chemical map for all different cells at a subcellular resolution at the whole organism level. And this chemical map is correlated with the metabolic features of those different cells. Then with this platform, then we can image animals at different age, with time. Then we found very interestingly, at a very early age, although those animals do not show any obvious difference yet, so their metabolic features in different cells already show drastic difference. So it really shows it's very important for us not just see the structural connections and changes, but it's very important to track the metabolic changes with time. So currently our deconvolution is only limited to 4 principal components. It's mainly because of noise. So I think with better denoising and deconvolution, it will allow us to decode more metabolic features from the SR spectrum. So I think the machine learning algorithm at the Janelia will help these areas. So this will particularly useful for imaging tissues with a high metabolic heterogeneity like the brain. The other technology we use to map metabolic changes is based on true photon fluorescence microscopy. So we use it to capture the autofluorescence signal from NADPH and FAD and then calculate their ratios. So then when the cells is more toward mitochondrial beta oxidation using lipid, we can see the FAD levels is increased as we detected in the intestinal cells, which is a fascial tissue of C. elegans. So this offers another way to track the balance between glycolysis towards lipolysis of oxidation. But this imaging platform will be also greatly benefit from better denoising, especially for 3D imaging using tissues. So I think in Gary's talk, he will tell us the really power of genetic engineered biosensors for metabolic imaging. So I think the molecular tool development at Janelia can help us develop more biosensors for different metabolic factors. And also, it's always better to have more than a hammer in the toolbox. So I think a platform to integrate multiple individual imaging modules for multiplex imaging will be very powerful. It will allow us to analyze metabolic changes from many different angles. In addition, I think this morning from Christine's talk, we can appreciate how imaging mass spec is involved in recent years to improve spatial resolution and also sensitivities. So it's really give us a lot of metabolic details. So the information we can get there is much more than any other imaging master we have. But most of the time we only see 2D image. So I think with the great success of Fly-EM at Janelia, it will be great to apply those 3D reconstruction platforms for imaging mass spec. I think it will allow us to really get a 3D metabolic imaging of the tissues and study its interactions with other tissues. And another challenge is on chemical annotation of mass spec data. So for example, if we look at metabolic data that we got from C. elegans samples, we often get 15,000 mass features, but only 2,000 of them can be annotated with exact chemical identity. And the same issues will also happen to fly, mice, or human samples. So, if we're comparing to genomics, so for 30% of genes, although we don't know their molecular functions, but at least we know their sequence and their gene ontology. But for more than 80% of mass data, actually we don't even have annotation, we don't know what they are. So I think this is a grand challenge if we really want to study metabolism in deeper manner. And again, I think the computational platform in Janelia will really help for these areas. And then especially with the multidisciplinary approach and also large scale platforms. So I really hope this can help us tackle this challenge. So overall, I think the strong expertise at Janelia on molecular tool development, imaging, and computation, we really promote innovations in metabolic imaging in vivo, and this can help drive for the cellular physiology studies.
That's wonderful. Thank you so much, Meng. We're now open for discussion, and I'll start with asking a question that I'm sure you've had many settings. But I think particularly here, what is, it would be interesting to the people here. What do you see as the time dimension and the spatial dimension of these technologies, including the hyperspectral Raman imaging and your 2-photon fluorescence microscopy? I think that's going to be really important for us to be able to scale between, organelle to cell to tissue levels. So where do you see, where are we right now in terms of how fast, for instance, in your worm, how many different time periods can you image, how smoothly, and what is the spatial resolution?
Yeah, so for the spatial resolution for both hyperspectral SRS and 2-photon imaging is a diffraction limited spatial resolution, but because both are using infrared laser, so it's a little bit worse than the visible laser. So it's around 300 nanometers. So you can still kind of get the information at the subcellular resolution. And in terms of the time scale, actually for 2 photons, it's very quick, but for hyperspectrum SRS images, the current limitation is if we want to span a big range for different wavelengths, the laser tuning will take time. So that's a main limiting factor right now. So we can get around 300 different wavelengths, about 5 minutes. So to scan the whole range. So therefore for worms, it's not a big issue, but I show you the brain image. And for that, we'll need about hours to kind of really get patches for the whole brain. So I think that's area I feel with a new instrumental modification, like to help the laser tuning faster. Well, it has been improved dramatically. Like the first scope that we built 10 years ago, it maybe take a day to get the brain image. So now, within hours, we can do that. So I think with more development, it will become faster and faster. Yeah.
That's great. Any other questions? I see that we're running a little bit past our time, so we'll hold off any further questions for Meng until the very end when we have a broader discussion. So Gary, please share with us your work.
Sure. It looks like I'm not on my first slide, so I'll go back to there. So first of all, I want to thank Jennifer, Vamsi, and Wang for the invitation to talk at this workshop. I think it's a very exciting an important set of subjects. And I already at this point in the day, it's going to be hard to cover completely new ground, but I'm going to talk about 2 sets of complementary tools that I think are very important for dissecting dynamic and compartmented metabolism, both in brain and in elsewhere. And so the brain isn't unique in having very dynamic, very compartmented metabolism, but it is kind of an extreme case, and it's extreme in a few ways. In humans, the brain consumes up to 20% of whole body energy, basically hitting about 10 times its weight in terms of energy usage. It's extremely dynamic, varies in seconds to minutes, and it's very spatially localized. So in seconds to minutes, one part of the brain versus another part of the brain becomes very active, and also it produces a lot of energy demand. Now, in addition, I'm not going to talk about all these things, but the brain metabolism is very incompletely understood. Lots of important problems remain to be solved. Compartmentation at both the cellular and subcellular level, the mechanism of these dynamic responses, the balance between glycolysis and mitochondrial metabolism, alternative fuels. Already you've heard a few people mention the ketogenic diet, and that's actually how I got interested in brain metabolism, because it can control excitability. The ketogenic diet is actually capable of treating epileptic seizures very effectively. And finally, wide open spaces in development, aging, neurodegenerative diseases, causative role of altered metabolism. So one obvious tool for addressing these questions is metabolic biosensors. They're really good at both dynamic and compartmented metabolism. And I will give you an example of 2 sensors deployed in an acute hippocampal slice from a mouse. We've excited these neurons with synaptic stimulation, and we follow these 2 sensors at the same time, these 2 genetically encoded sensors, using fast fluorescence lifetime imaging, which is particularly good for quantitation. And what you see is when the cells are excited, the calcium goes up very briefly, and then the cytosolic NADH to NAD ratio goes up for several minutes, reflecting the metabolic response of the tissue. Now, we don't just look at these changes but we can tie them in very directly to metabolic pathways in the cells. You've been hearing a lot about glycolysis. And at baseline, in the baseline state of the brain is very good at complete oxidation of glucose to carbon dioxide. So it goes glucose to pyruvate and is used in mitochondria. But when cells are stimulated at the peak of this NADH response, there is apparently excess glycolysis, there's excess lactate in the brain. And one possibility is just that neurons are doing more glycolysis, they elevate their NADH, convert pyruvate to lactate. But another that Jared mentioned earlier is this idea of an astrocyte neuron lactate shuttle, which has been very popular over like the past 25 years. And that idea is that astrocytes have their glycolysis stimulated, they produce lactate, export it to neurons, which then use it as fuel. So this NADH response in the cytosol of neurons could come from either of these things. We can test this with pharmacology, a nod to Mark, and if we block the monocarboxylate transporters in the surface of actually both neurons and astrocytes, we should eliminate this signal if it's from the astrocyte neuron lactate shuttle. So that's the prediction. And we have independent evidence that this blockade is effective. But what we in fact see is an even bigger transient in NADH to NAD, probably because the lactate produced by the neurons can't escape the cell. And so it gets bottled up and it pushes back pressure on this pathway. So this is an example of how we can use metabolic sensors to look at this cellular specific, and also I should mention compartmental specific localization of metabolites, and also address specific hypotheses with it. Now, metabolic sensors in general give a quantitative readout of metabolic state. I mentioned that fluorescence lifetime imaging is not the only way, but it's a very good way of correcting for sensor expression level. We can get single cell or subcellular resolution, basically optical resolution, and we can even target these genetically encoded sensors to specific subcellular compartments. And we can do fast imaging, not just in live tissue, but also in vivo. Now, we can use several of these sensors, but we need to engineer a sensor for each target. And other speakers have mentioned the frustration that there aren't sensors available for everything yet. And I'll come back and address that at the end. I think that's one of the major challenges. We're also limited by color to 2 to 4 simultaneous analytes. So we can measure a few things, but we'll always be a little bit frustrated in terms of defining what exactly is going on with the metabolic pathways in these cells. And so a complementary tool that's also already been mentioned in the workshop multiple times is the use of mass spectrometry imaging. And because of the mass spectrometry, it has a deep chemical dimension. We can measure many metabolites simultaneously. And we get the spatial dimension by scanning. And Tara also already described this. You basically, you can use a laser to desorb and ionize, get a mass spectrum. Each peak is a different chemical compound. And then if we scan the laser across the tissue, we can construct images for each individual metabolite peak, and we can quantitate the metabolite in basically each pixel of the sample. Now, the challenge is the current spatial resolution is about 5 microns, and that's good enough to get a layer of relatively homogeneous neurons, but not good enough to get isolated astrocytes or oligodendrocytes in the tissue. And so that's an area for improvement. A challenge is to get finer cellular and even subcellular resolution. And there are good physical mechanisms by which this might be accomplished, but it will take a concerted effort. Second, and Josh already brought this up, he showed very beautiful data on a temporal component of mass spec in vivo. And we've been working on getting this really fast, seconds to minutes range in ex vivo samples. And so we use acute brain slices. This is a very standard preparation for studying neuronal physiology, brain physiology. It's not quite as good as in vivo, but it's way better than culture. And we use it almost immediately out of the animal, and the physical structure is conserved. And then we can put this in a physiological chamber where we can stimulate either with an electrode or with optical excitation or with chemical excitation. We can image fluorescently while we're doing that, and we can perfuse C13 fuels. So very good manipulation. But because mass spec imaging is not live cell imaging, we need fast preservation, which we do with a flash heat, flash freeze using fast thermoelectric changes in the temperature. And then we can thin section and put these in the cryotome for mass spec imaging. So here are some results. Here are two sister brain slices from the same mouse. And we put electrodes into both of them, but we electrically stimulate only one of them. And we do that for 30 seconds and we flash preserve 30 seconds later. And what you can see with the mass spec imaging are the molecular changes. So we see just in the region that's been stimulated, we see a decrease in the level of ATP and a corresponding small increase in the AMP in that same region. Now, this is just the tip of the iceberg of the information we can get from an experiment like this, because we can, all the metabolites in green here, we can assign, annotate peaks for each of these metabolites and measure their changes. On top of that, we can put in C13 fuel molecules and actually measure the rate of incorporation from which we can learn the pathway fluxes. So this is just one metabolite, but again, we get many, many of these. So we think we can meet this challenge of the very fast temporal component in this ex vivo prep using fast manipulation and fast preservation. We can use stable isotope labeling to learn the fluxes. And, as we were discussing before, this is very challenging to do by any other means, but with the mass spec imaging and with C13 labeling, for example, we can do this. So I think these two approaches are extremely complementary. Each of them is kind of unbeatable in its respective area. So with the metabolic biosensors, we can image fast, we can image living tissue in vivo, and with the mass spec imaging, we get this deep chemical dimension and fluxes. So the big challenges, I think, are in the area of spatial resolution for the mass spec imaging. And I think we can push that to the cellular and maybe even subcellular level. And then my hope is that from this systemic view that we get from mass spec imaging, we can decide, well, which sensors are really worth making. We have to go back and engineer them each, but we can learn what are the harbinger metabolites from the mass spec imaging, and we can engineer sensors for each target. And we've recently completed the development of a very high throughput, high content screen for both chemosensors, and if we have a chemosensor for chemo effectors, like the one I was talking about before, based on Vamsi's LB-NOX enzyme, and this screen is enabled by microfluidics. So I think these are the two very good complementary techniques that address different sides of this multi-scale nutrition and metabolism question that are really worth pursuing. And I will just thank my collaborators, both inside the lab and outside the lab, for the mass spec imaging work. We've been particularly dependent on Dr. Natalie Agar's lab at the Brigham and Women's Hospital. They've been enormously helpful and great in developing that project. So I'll leave this slide up there for just a second. I'm happy to take questions.
Fantastic. I'm just looking at the question answer. Yeah, so we don't, please, oh, wow, we got some people from our panelists. So Vamsi, why don't you start, and then Ralph and Lou, you can put out your questions.
Great. Gary, that was wonderful. So on the previous, on the penultimate slide that you just showed, you said that improving that spatial resolution is that challenge that ought to be tractable. And I think Tara a little while ago pointed out what some of the challenges are. So can you just elaborate on what, do we need innovations in mass spectrometry, or are those sensitive enough? Or do we need innovations in the excitation, or do we need new chemistries for the MALDI? I mean, can you just elaborate on what the path?
Sure, I think there are promising things in all those arenas. So as Tara mentioned, just laser spot size can be improved, things like that. But sensitivity, as you mentioned, or someone mentioned before in Tara's question, is a big issue, and there are tools for improving that. There are mechanisms for improving the efficiency of desorption and ionization. There's the possibility for extremely high spatial resolution of abandoning MALDI, so not using a matrix and using an ion beam instead. And there the challenge is balancing the tendency of high energy beams to fragment the molecules with the ability to detect them. But there, in principle, we can get much smaller, much finer spatial resolution that I think could approach the ability to, get a few pixels per mitochondrion instead of a few pixels per cell.
Great, Ralph.
Really nice, Gary. Just maybe to follow up on that question, it seems to me like another challenge is not simply the increased resolution of the mass spec imaging, but being able to integrate information about the tissue in the same image, so that you're not left with trying to get information about tissue architecture from one slice and then information about metabolism from the next slice. What needs to be done to make that happen too?
So we're looking at that right now. There are good methods now, just very new methods. We're collaborating with some people to label antibodies in ways that can be measured in the mass spectrometer, so we won't have to use sister sections the way that you've described. And then our hope is to get metabolic signatures of the different cell types that we can use even without the antibodies. So that's how we're hoping to push things in that direction.
Great, thank you. So this would be like a CyTOF, only it would all be done in Cy2.
Exactly, and it wouldn't be a separate instrument from as the CyTOF is. Right. And as I say, we want to go as quickly as we can from getting label registration with metabolism to just using the metabolic, to basically using the multidimensional metabolic information to tell us which cell types are represented in a particular pixel.
Thanks, Gary.
Last question from Lou Cantley. Lou, you have to unmute.
Sorry. Yeah. Gary, you started out by mentioning epileptic seizures, and I'm wondering whether you're taking this, going to try to do ex vivo epileptic seizure analysis. You pointed out that ketogenic diets were developed to treat that disease, which is true. And, there are two major ways of getting epileptic seizure syndromes. One is tuberous sclerosis, where the two TSC genes are defective, and you get too much TOR activation. And the other turns out to be mosaicisms in PIK3CA and PIK3CA and AKT in that pathway. So I've always been under the assumption that the ketogenic diet works because it lowers insulin. But it would be terrific to test that out in an ex vivo system where you have control over everything.
Sure, and that's exactly what another part of the lab is doing, Lou. We look at ex vivo seizure activity, which we can induce with the chemoconvulsant in vivo, and we know that in that case, of course, we're just changing fuel molecules, or we're just using bad knockouts versus not bad knockouts, and so it's ex vivo. There's no change in insulin signaling, and we see that KATP channels, which are another of these sensors, sort of like the nuclear receptors and metabolites, they are a membrane ion channel that is a sensor of ATP and ADP in the cell. We know that they're probably responsible for the altered excitability and the altered seizure activity. So I know your focus on insulin, and we've thought about that a lot. I think it's something we can address in animal models, but we have independent evidence that these ATP and ADP sensitive channels and the altered cellular fuel metabolism is likely responsible. But I think that will probably depend on the model. I think the exact answer will depend on the model.
Good, thanks.
Great, really great discussion. We've got to move on. Thank you, Gary, for a really wonderful presentation. We're going to now move to Daniel Hochbaum. Daniel, you want to share your slides?
Can everyone see my slides?
Yes.
Yep.
Yes,
great.
We can't see you though.
Can't see me.
That's good. Yeah, now we can see you.
Awesome. Okay. My name is Daniel Hochbaum. I'm from Bernardo Sabatini's lab. I'm a postdoc in the lab, and I'm coming to this meeting from a bit of a different perspective. So forgive me for any nomenclature or anything that's unclear. I'm happy to clarify in the question and answer. So we often think of ourselves as individuals observing our environment, making internal decisions based off of the information we sense, and then producing appropriate actions. This perception of ourselves implies that our decision-making, our behavior, and who we are is informed by our environment, but is somehow distinct. But in many ways, this is not true. For example, the time of day drives our internal circadian rhythms, dictating dramatic changes in our behavior, such as sleep. For many animals, the time of year activates internal seasonal programs, including that induces very different modalities of behavior. For example, hibernation in the winter and active hunting in the spring. So in many cases, it's not just that our environment informs our decision-making, but instead our environment directly shapes our internal decision-making processes and dictates the behaviors we engage in. And in the wild, some of the most striking changes in animal behavior are accompanied by changes in metabolism. For example, these are African striped mice that live in a semi-arid environment in South Africa. During the dry season, when resources are limited, they live along this riverbed, which is sheltered and has sufficient food sources, and their metabolic expenditure is minimized. But during the wet season, high caloric annuals grow in these adjacent plains, and the mice expand their home range territories and shift them away from the riverbed towards the plains to forage, and their metabolism ramps up. So this is illustrated here, where the gray zones indicate individual animals' home ranges during the dry season, and the black is their home range during the wet season. And as you can see, the wet season ranges tend to be larger and shifted towards the plains. However, this shift does not come without risk, as these plains have less coverage and the animals are more prone to predation. So what are the signals these mice integrate that mediate these exploratory behaviors? In a follow-up study, they found that during dry season, the animal's level of thyroid hormone, which is a critical regulator of whole body metabolism, was low. During the wet season, their metabolism ramps up, corresponding with an increase in thyroid hormone levels. So this is suggestive that perhaps thyroid hormone could not only be involved in changing metabolic state, but also could be a driver of these exploratory behaviors. Similar observations have been made in multiple species, including primates. Here are male gray mouse lemurs from Madagascar, and they have a huge surge in thyroid hormone levels during their breeding season, which again corresponds to nutrient-abundant wet season. As a result, they increase their metabolic rate, they eat more food and consume more oxygen, and like the striped mice, they greatly expand their home ranges between the dry season and the wet season. So what these and other studies guide us towards is a framework in which resource availability concurrently regulates metabolic expenditure and exploratory behavior. For example, during times of plenty, it's advantageous to increase energy consumption by effector organs like muscles, to sleep less, to explore more, and to take more risks to gain access to food, mates, and other resources. Conversely, when resources are scarce, it's advantageous to become more withdrawn, to sleep more, to explore less, and to reduce your overall metabolic expenditure. However, if this program is engaged in inappropriate times, it will manifest in pathological conditions at the extremes of this axis, such as mania and depression, which are common psychiatric presentations in humans with hyper and hypothyroidism, respectively. So in sum, the ethological studies and human disease evidence lead us to a hypothesis that thyroid hormone directly exerts parallel changes on peripheral organs in the brain to coordinate energy expenditure with appropriate exploratory behaviors. So to test this hypothesis, we developed a simple mouse model of hyperthyroidism, where we bypass the exogenous regulation of thyroid hormone and simply inject animals with the thyroid hormone T3 or vehicle control. And over the time course of a few days, we look at changes in RNA profiles, protein levels, and functional connectivity of neural circuits, as well as behavior. We first used indirect calorimetry to validate changes in metabolism. This is a home cage system where oxygen consumption and CO2 production rates are continuously monitored. After about 2 days of treatment, we start to see increases in oxygen consumption, CO2 production, and overall energy expenditure. And in addition, these home cages allow us to monitor course behaviors. And what we see is that hyperthyroid animals eat more, they increase their food intake, and they also move around the cage more. They're hyperactive relative to controls. So to get a finer resolution of how the animals actually change their behavior with thyroid state, we turn to a paradigm that mimics some aspects of the field studies I described before. In this elevated plus maze, which we can do in the lab, there's an enclosed arm and an exposed arm on an elevated platform. Mice find the exposed arm naturally aversive and spend most of their time in the enclosed one. We asked whether we could mimic some of the observations made in the wild by purely manipulating thyroid hormone levels to see if animals become more exploratory of a perceived risky environment. So we tested mice after 3 days of treatment, and we found that indeed, hyperthyroid mice spend a larger fraction of the session on the exposed arm, suggesting hyperthyroid mice engage in more spontaneous exploratory behavior. So the natural next question to ask is, how does thyroid hormone induce these altered behaviors? To address this question, we noted that the receptors for thyroid hormones are ligand-gated transcription factors that activate transcription of genes when bound to T3. These receptors, of which there are two, alpha and beta, are expressed in hypothalamus, which is expected because hypothalamus is known as a key regulator of thyroid hormone levels. But additionally, these receptors are expressed highly throughout cortex. So we wanted to focus on higher order cognitive behavior. So we decided to perform single cell RNA sequencing of a region called secondary motor cortex that's implicated in action selection and planning. When we compared the transcriptional profiles between control and hyperthyroid animals, we found that many cell types listed here on the vertical axis changed their expression of tens to hundreds of genes. As a class, excitatory projection neurons differentially express the most genes. And when we looked at what biological processes are associated with these induced genes, we found a very strong enrichment for genes associated with axon development, genesis, and guidance. So taking a closer look here, I've plotted some representative thyroid-regulated genes and excitatory neuron cell types, which are listed along the horizontal axis. The dot size is related to the percentage of cells we found the gene expressed in, and the colors, the degree of induction between control and thyroid state. We have induced genes on the vertical axis, and these include presynaptic molecules such as norexins and semaphorin, auxiliary calcium channel subunits, and at the bottom here, we have canonical thyroid control genes. I could spend all day on this slide, but for the purpose of this talk, I'll single out a single gene, ROBO3, which is the top upregulated gene in excitatory neurons in the hyperthyroid state. ROBO3 is a single pass transmembrane protein, which has been extensively studied in the context of development, where it's critical for axon guidance. So given the induction of ROBO3 and axon guidance pathways more generally in cerebral cortex with hyperthyroidism, we hypothesized that changes in synaptic connectivity and function could occur. So we targeted neurons with channel rhodopsin that we know induce ROBO3, and we developed a protocol to titrate the amount of blue light stimulus, which was useful for parameterizing aspects of direct excitatory currents and indirect inhibitory currents, which I'm happy to discuss in more detail after the talk. The upshot is what we found was that hyperthyroidism modulated both excitatory and inhibitory drive onto excitatory neurons, increasing overall excitation relative to control, but simultaneously sensitizing indirect inhibition at low light levels. What's more is that these differences required the presence of ROBO3. As you see here, knocking out ROBO3 prevented these changes from emerging between the control and the hyperthyroid conditions. Finally, we wanted to know what aspects of metabolism behavior are dependent on ROBO3 induction. Our first attempt at this was to introduce a systemic version of CRE that is expressed in neurons, or a truncated version of CRE known as delta CRE, which is inactive throughout the brain of ROBO3 conditional knockout mice. We injected virus into these animals and then ran them through the same metabolic home cage system I described before, first treating both cohorts with vehicle for 5 days and then treating with T3 for 5 days. We compared the final 48 hours of the vehicle period and the final 48 hours of the T3 treatment period when metabolic and behavioral effects of T3 are saturated. And what we found is that both the control and Robo3 knockouts have robust increases in metabolic rate when treated with thyroid hormone. So Robo3 is not a regulator of the metabolic changes driven by thyroid hormone. The Delta Cree control had the expected behavioral phenotypes of increased food intake and increased locomotion. But remarkably, while the Cree-treated animals maintained their hyperphagic phenotype, they continued to eat more, which is a phenotype likely mediated by the hypothalamus. They no longer increased their locomotion after T3 treatment. So to summarize what I've shown you so far, these have been little vignettes of the experiments we've been doing. We've seen that hyperthyroid mice have expected increases in their metabolism. But in addition, thyroid hormone acts locally in cortex to activate axon guidance programs in cortical projection neurons. And this results in rewiring of these projections. The mice become hyperactive and exhibit increased exploratory-like behaviors. And critically, we found that this circuit rewiring and change in locomotor activity require the induction of Robo3. So what I hope I've demonstrated for you today is that thyroid hormone not only functions as a critical regulator of metabolism, but also directly modifies brain circuits to shift behavioral tendencies to match metabolic needs, driving individuals one way or the other along this axis of behavior. And I'll stop there and take questions.
That was great, Daniel. Really terrific. Super exciting. I'll start by asking the question, what does thyroid hormone do to other organs in the body besides the brain? And
I'm sure, yeah, I'm sure other people have much better answers than I do, but it's involved in lipid metabolism in the liver. And I think that's, and also it encourages burning, heating of fat cells.
And how does it intersect with the insulin regulation? How are the two? I've been hearing a lot about how insulin is a major metabolic regulator, and now being introduced to the concept of thyroid hormone as
actually also, I have
so are they yin yang? Are they working together? How do they?
I actually have no idea about the interaction of thyroid hormone and insulin. That's a great question.
Other questions from people?
Maybe I can ask one.
Yeah.
I'm wondering, like, this is a beautiful example that thyroid hormone can orchestrate a lot of responses, and maybe they function as a package. So the Robo3 is basically saying what is responsible for the behavior and neuronal activity. And how about that you take a similar approach, you take one like a peripheral metabolism element out and see if the neuronal function will be affected.
Yeah, that's a great idea. I would be very open to ideas of which to take out. You're saying that with a direct change in metabolism, would it produce similar changes in connectivity and so forth?
Yeah, I was wondering if the behavior change is dependent on metabolism.
Yeah. Well, what I can tell you is that when we do naturally occurring changes to metabolism, like if we starve the mice and so forth, we can activate similar genetic cascades. Now, we think that that is directly downstream of thyroid signaling, but it could involve other metabolic pathways as well.
Fantastic. Okay, so if I don't since we don't have any immediate questions for Daniel, let's move to our discussion. And so for that, Daniel, if you could stop sharing and all of the panelists can come into view. Wonderful. So yeah, we've been hearing a lot of different aspects of different approaches for imaging responses through, whether it be, mass spec or to photon, the Raman approach. But then we've also heard from Daniel about how a very specific molecule, thyroid hormone, can really drive all kinds of processes. And so I'm curious as to how, Gary, you and Meng, what's your perspective in terms of the role of single hormonal elements driving a lot of changes? Do you see evidence for that in your systems that you're looking at?
Well, we haven't really taken this on yet, Jennifer, but yeah. I think that you get what you see. So I come from a more retail approach, with the biosensors, looking at individual molecules in very well-defined paradigms. I anticipate that as we go to this more systems level view, where we see lots of things, we're going to see unexpected stuff. And, but as far as like Dan's type of experiment, I think it just, it's why we, in addition to having the visualization tools, we need the experiments to do with them, and so, we've done neuronal stimulation and, various pharmacological manipulations, but we could look using the same tools. We could look using thyroid hormone presentation or other things. So I think, these enabling technologies are not the end, they're the beginning of, how we approach experiments.
Right. But I'm just curious as to, and Meng, you could maybe address and expand on this concept of whether there are key molecules that we should be focused on in terms of crosstalk between the brain and other organs for this inter-organ communication system. Clearly, insulin's playing a really important role. I think, Dan, your work is pointing to thyroid hormone. Are there other molecules that we as a field should be sort of paying attention to, or do people speculate one way or another on that? So Meng, do you have?
Yeah, so our example, actually, I went through quickly, is a lipid. So it's a specific lipid molecule. So I think they are well known for building blocks and energy fields, but I think there are more and more studies showing they can serve as a single molecules in a ways like cytokines. And then so they can cross different cells and different tissues and to communicate the physiology between different parts. So the particular example we show is from the peripheral to neurons. So in worms, it's easier. You just need to secrete it into the body cavity. Then they can target on neurons long range. But in brains, we know they're like, I think the field knows some of the lipid need a special carrier to cross the blood-brain barrier. So actually there you have additional regulations. And then from there, they will also have a carrier to go to different places. I think very similar to the steroid stories told by Daniel. So different receptors in different neurons may mediate the downstream effect on different type of like neuron astrocyte or microglia.
Great. We have two people from the panel, Daniel and then Chris. Daniel.
Yes. So I just want to add to, it's very important to identify these factors, right, that regulate metabolism. But I think the effect these factors have is a combinatorial factor. And I use as an example insulin and glucagon. the effect of these two hormones is depending on the ratio. If insulin is high, then insulin signaling dominates, but glucagon is still present, and vice versa. So I think identifying the factors is important, but then knowing what is the ratio of these factors, right, is what really determines the effect in the body, I think.
Mm-hmm. Chris?
Yeah, I would just expand upon that to say that any consideration of metabolic regulation has to include the endocrinology. All right, so this was the thyroid hormone talk was lovely. I agree with Daniel that insulin and glucagon ratio is important, but we've now begun to understand, quite surprisingly, that glucagon secretion from the alpha cell is required for normal glucose-stimulated insulin secretion from the beta cell. So there's paracrine communication from the alpha cell to the beta cell that enables the beta cell for insulin secretion. So there's a set of dynamics with the in terms of intertissue regulation, as you're asking about, Jennifer, where we would ignore the endocrine factors at our great peril.
Mm-hmm. And do you think that these endocrine factors, I mean, one of the things that I think was especially exciting about Daniel's talk is the ability to drive the response of the tissue and the organism to different circadian cycles, food availability cycles, et cetera. It's almost as if it's working, in the case of the thyroid hormone, it's working at a organismal environmental level. It's connecting the organism to the environment at some level. Which
Sure, but you could say the same thing about leptin, ghrelin, NPY, et cetera, et cetera. So I think we have to look more broadly and more harmoniously at the endocrine milieu if we're going to understand the specific functions of metabolites and signaling.
Fantastic. Wang?
Yeah, I want to follow up on this question about how the metabolism could change cell behavior. And I probably will scale down from what Chris just mentioned at the system level. And so I'm more thinking about the like intracellular and subcellular level. So some mechanisms propose how metabolism regulates cell function is like it regulates epigenetics, it regulates protein modifications. But just think that a lot of metabolism occurs in mitochondria. We're assessing the cellular metabolites as a whole cell. We actually don't know that those metabolites that we think modulate epigenetics are actually present in the nucleus, where the target is. So we need tools that could resolve those metabolites whereabout and the concentrations across in different subcellular compartments. I think right now looking at the tools, mass spec still has way to go because it's not even at a single cell level yet. But the biosensors has a good shot. I know Gary has done a lot of those things, and you're looking beautifully how the NAD/NADH ratio changes in the cytosol, but can you look how it changes in the nucleus?
Absolutely. Nuclear targeting is about the easiest thing we can do with the sensors. So
yeah, and also, the other metabolites like acetyl-CoAs and alpha-ketoglutarate, it's extensively studied that has a lot of function, but we just don't know where it is.
We don't have sensors for those yet, but we're hoping to be able to enable those sensors to be made.
Vamsi, do you have you were going to make a comment?
Thanks, Jennifer. I was going to make one comment, and that's, the title of this last session was quite provocative. It was, I think, something along the lines of Beyond Connectomics, but I would love for the panelists and the student speaker from this last session to maybe just comment a little bit about how these two things could be directly connected. Let's not get beyond the connectome, but for the neuronal connectome project, is there an opportunity to somehow layer on metabolism there? So when two neurons are touching each other, for example, is their metabolic state somehow related to each other as well? So I would love to ask the panelists and student speaker to maybe comment on connecting the connectome to metabolism.
Great question.
I think the challenge is spatial resolution, right? And I mean, if you knew exactly where you wanted to, what junction you wanted to look at, and you could target a sensor there, then you'd have an entree to that question. But to do it in a less targeted way really requires that we have much better spatial resolution with the MS imaging.
I'm trying to think some of the hyperspectral imaging we are getting from both brains and from worms. So for like worms in the, they don't have a brain, but around the neuron connections, I think we do see some heterogeneity among different neurons. And we don't have the resolution to look at really the synaptic junction, but from the two neurons that are connected. So actually you can see heterogeneity there. I think the interesting thing will be to couple with optogenetic tools to really changing one neurons and see actually how the metabolic status in the neighboring neurons is responding to that. And for brain, actually, because the time restriction just mentioned to Jennifer, we cannot do those dynamic like in the brain slice yet, but hopefully with a faster imaging platform, we can do the same thing, because we do see at the snapshot among different neurons and astrocytes, the metabolic features are very heterogeneous. And then whether or not they're related to their neighboring neurons or neighbor astrocyte, we don't know. We have the resolution to do that. Just right now, the time scale is not ready.
I think in this morning's liver session, right, we discussed as a group organelle crosstalk, and I saw a paper in the bioRxiv. I think it was posted by Sebastian Sung's group from Princeton a couple of months ago, where from the existing connectomics data, they were actually able to reconstruct things like mitochondrial connectedness as well. And so I just wonder, I don't know if any of the folks here are sort of professional connectomics folks, but I wonder if at least the reconstruction, Gary, if some of the direct metabolite measurements are hard, maybe reconstructing the localization and morphological properties of organelles may be easy potentially.
I think that would obviously be easier than the metabolites. Yeah, I think that's probably achievable, especially with the EM level connectomics. I think you would, if you look for it, it's probably in a database you can already look at.
No, and I think that's exactly what Sebastian Sung's group did. They actually took existing connectomics data sets and sort of reconstructed organelle morphologies.
Well, in particular, the ketogenic diet that you talked about, how you saw huge differences in these different parts of the brain, that signature. But I think the other thing, Gary, that you mentioned, that you felt that you could see a mass spec signature that could distinguish neurons from astrocytes or glia. That I think is going to be extremely important, because those cells are incredibly supportive metabolically to neurons, and their distributions, their activation. For instance, when a neuronal circuit gets activated, could make a huge difference.
Yeah, I think that's going to be tremendously interesting. I think there's good reason to hope that we can get such signatures. There are even in MR spectroscopy in vivo, there are peaks that are considered to be diagnostic of either neuronal or astrocyte metabolism. And so, I think most of those things, they're not 100%, but I think because we have this very multidimensional picture, I think the idea that we'll be able to look at the principal components that distinguish them and use that to segment the cells, I think that's not far-fetched.
Just a quick question before we get, Lou's just raised his hand, but I want to ask whether anybody has been looking at glycan, sugar residues on proteins, carbohydrate modifications.
Yes, yeah, I think people are definitely trying to do that by mass spec. If you pre-digest, so my collaborator Natalie Agar has been using enzymes to cleave off the glycans, and then they can be visualized by mass spec imaging. Yeah.
Meng, have you've been in that arena too?
Yeah, so we are using also mass spec to do that. But for imaging, direct imaging, we have to label the sugars with deuterium first, then we can trace their dynamics. But for endogenous sugar, we cannot do that yet.
Right, because those sugar, the type of sugar that's put on can be very much dependent on the metabolism, you know, what sugars are being made. So let's quickly, Lou's had his hand up for a while. So Lou, do you want to?
Yeah, just two questions. First, whether one way you could do cytosolic versus mitochondrial or other compartment metabolites is not at the single cell level, but at a tissue level. It's just do digitonin permeabilization, which will pop a hole in the plasma membrane within seconds and allow you to see what leaks out. Anything that leaks out on that time scale is going to be in the cytosol. But the question is, can you do it at a more sophisticated and single cell level with patch clamps? You know, patch clamps certainly allow you to measure potassium and sodium levels in the cytosol, but might it be adapted? Has anyone tried to attach adaptive patch clamp to measure metabolites?
Yeah, their Nature, I think last month, their Nature Master paper, they actually even patched the single lysosomes and do metabolomics analysis there.
Yeah, cool.
That's a great idea.
Yeah, I'd love to see that paper. Maybe you can send it to me.
Yes, I will.
Yeah, that would definitely be fascinating to see at a single cell level, even different parts of the cell, dendrite versus axon areas. Okay, other questions? Well, actually, there's one last. Daniel hasn't told us what his view was in the question that was posed from Vamsi in terms of the connectomics and what you.
Yeah, so I come from a very different perspective, but I think the point of this project that I just presented to you is that these factors that are influencing metabolism in other parts of the body also feed back directly on how neurons are connected to each other and
how they develop. Yeah.
Right. Yeah. And so whether that is, it's not necessarily a change in those neurons' metabolism, but it's a change that is regulated by whole organism metabolism, presumably for evolutionary benefit, right? You need to rewire your circuits to take advantage of your current metabolic state.
That's fantastic. That's a really great concept. Just how the whole system is feedbacking on itself, especially at the level of the brain. Okay, I think we're running out of time, and I just want to thank Daniel, Meng, Gary, really for a fantastic set of presentations, and also the panelists for really great discussion. I'm going to now hand it over to Janine, who's going to lead us into our next mode.
Yes, perfect. Yes, I just want to echo Jennifer. Thank you all for a great session 4, and to all of the speakers and discussion leaders for a really enlightening day 1 of the workshop. I want to thank the audience for joining us today also. This concludes the open portion of the workshop, but please come back and join us for day 2, which starts tomorrow at 11 a.m. Eastern at the same link. Panelists, please don't go far, but we will take about a 10-minute break, and then we will meet for a closed discussion for about 40 minutes. And so I'm about to send an email just with a link reminder of where you can join, and we will see you then. Thank you.
Thank you so much.